Okay, so these next two chapters are both on host defenses, so it's broken up into two different sections, um, mainly by topic, which you'll see once we get through them. Um, these chapters, there's going to be a ton of overlap with your other classes, from anatomy to pathology to even other clinical classes. So there's going to be a ton of information that you will have probably already seen or known, and you will certainly see again in other classes. So this is something you'll definitely want to pay attention to. And it obviously directly relates to microbiology because of the interactions between microbes and our bodies. So this chapter is called Host Defenses. Um, and what that is basically the defense against pathogens. And there are multiple ways that our bodies defend against these pathogens. So, uh, as this says here, innate, non-specific protections. Innate just means you're born with it, or specific, what we call immunities. And we'll go through what all of these are, but of course, your immunity is your body's ability to recognize and fight off specific types of infections. So these immunities we have broken up into first, second, and third lines of defense, which we're going to mostly cover in the uh, next chapter, chapter 13. But here's a brief overview. So first line of defense is any barrier that keeps things out of the body. And some of these are obvious barriers, like your skin. That's considered a first line of defense. But some of them are not quite so obvious, and we'll go through a whole list of what those are. So they just limit the access to getting inside of your body. So a lot of times, for example, some sort of a microbe can live on your skin and not cause any problem whatsoever. But if that same microbe is able to colonize under the skin somehow, then you have an issue. So this is not considered to be an immune response because it's just it's like a wall. It just keeps everything out. Very general in action. So the second line of defense is the system of cells and chemicals that protect us by resisting pathogens. So inflammation and phagocytosis are parts of this because inflammation is a generalized reaction. Phagocytosis is of course, um, ingestion of foreign particles or uh, cells by cells called phagocytes, such as macrophages and whatnot. But they don't necessarily recognize any type of a specific pathogen. They just consume anything that is foreign. So these are both local and systemic. Local, obviously, being in one place. Systemic, meaning the whole entire body. And of course, this is only after the first line of defense has been breached. And then that brings us to the third line of defense. And again, don't be too concerned about these right now. We're going to go over all of these in detail. We're just doing an overview right now. And this is the acquired immunity that we get once we have been exposed to something. We actually have memory. Well, we don't, but our cells have memory. Our lymphocytes have memory. Uh, these B and T lymphocytes that we'll get to in the next chapter. And that way, once they can recognize them, they're more efficient at killing them the next time you're exposed to them. So different microbes will potentially give different reactions and different types of defense tactics because you're not necessarily going to need the same type of tactics for every single pathogen. And our third line of defense produces long-term immunity. in theory, your immunity will remain the rest of your life. It's not always the case, but we will oftentimes continue to produce memory or antibodies to something that we've been exposed to for 60, 70, 80, 90, 100 years, something like that. So most of these defenses overlap. Um, it's not like, one pathogen is only fought with, one of these defense systems. It's most of the time it's all three. So survival of a pathogen is unlikely. We're exposed to pathogens by the millions every single day. And so we are constantly fighting these things off, and obviously we don't get sick every single day. And if you're healthy and you have good nutrition, vitamin D from sun exposure and things like that, it's actually very uncommon for most people to get sick. Okay, so this chart right here, which is 12.1 in your book, just sort of breaks down graphically what we're about to talk about for the next two chapters. So under the host defenses, we have our innate and non-specific defenses. So these are the ones that don't remember, right? And under that, we have the first line of defense, which is just our barriers. So these can be either anatomical, like your skin, or physiological, like certain chemicals. For example, chemicals on your skin, like the acidity or the sebaceous secretions that resist colonization of bacteria and fungi and things like that. So that would be physical, chemical, and also genetic components. And this is just the fact that certain, seems like we talked about this in a previous chapter, but it's just the fact that certain diseases don't affect all animals, right? Like, humans don't get parvo, but dogs do. It's just because the virus isn't compatible with both species, and that's just the genetic makeup of it. So that brings us into the second line of defense, which are cellular chemical systems that are activated if the first line of defense is breached. And these are things like phagocytosis, inflammation, fever, which just increases the heat in your body and makes it hard for replication of pathogens, and certain antimicrobial proteins like complement that we're going to talk about in probably the next chapter. So that's innate or non-specific. And sometimes they're going to call these innate immunity or non-specific immunity. That's technically not true. It's not immunity, but just go with it. So acquired or specific immunity is a completely different story, and this is all under the third line of defense. And this is where we create memory to very specific pathogens. So we can get this specific immunity or acquired immunity two ways, either actively or passively. Actively is with an infection. You actually get exposed to it. And by the way, these can be artificial as well, such as in a vaccine. We'll get to that at the end of chapter 13. Or you can get them passively, and this would be from maternal antibodies, either through the placenta or through the breast milk, which would be IgA and IgG for those. And this involves B cells, T cells, which are both types of lymphocytes, as well as the cells that come from the B and the T cells, like cytotoxic cells, memory B and T cells, plasma cells, those sorts of things. Okay, so for this chapter, though, we are going to focus on first and second lines of defense, and for chapter 13, we will continue on with the third line of defense. So, as we said, part of this first line of defense are barriers. So these are inborn non-specific defenses divided up into three types, either physical barriers, chemical barriers, or genetic barriers. So all these things do are they just set up walls and just make it difficult for pathogens to come in and colonize. So this is just a list of physical, or what sometimes are called anatomical barriers. Your skin. So on your skin, the most outer layer of the epidermis, which is the most outer layer of your skin, is called the stratum corneum. Strata means layer. Well, stratum means layer. Strata means layers. Corny means like hard or tough. And that's just because in it is this chemical. It's a protein called keratin with a K, not to be confused with carotene with a C, which is a pigment made from vitamin A. Anyway, it says it's impervious. That's obviously not true, but it certainly makes it difficult for things to break through it, right? It's waterproof, and so that helps us keep fluids in and out. And this one is the layer that's constantly sloughing off and makes a lot of what is the dust in the air. And when it sloughs off, it evidently takes microbes with it. And along with this, part of this outer layer of your skin, not the stratum corneum, but the epidermis, are these structures such as your hair. Even though they're in the dermis, they're considered to be epidermal tissues. And occasionally these your hairs are periodically shed. And the term they use for this is desquamated or desquamated. And so D means like opposite or against or something like that. Squam just means flat. So I'm not exactly sure where the translation comes from, but your cells of the epidermis become what are called squamous cells because they're flat. And the hair cells are sort of similar to that because they actually die and just what's left over is the keratin. Anyway, it just means that your hair is shed. When you sweat, it also sort of washes the microbes out of the sweat glands and off of the surface of your skin as well. So the next barriers are your mucous membranes. Now remember, mucous membranes are the membranes, they're epithelial tissues that are in cavities that open to the environment, which would be your GI tract, respiratory tract, genitourinary tract, and even your eye, like your tear ducts and that. So mucus keeps these microbes from being able to penetrate through it because it's just a layer of mucus, and keeps them from being able to attach to the actual epithelial tissue. So when we blink, we produce tears, and also in the tears there's an enzyme called lysozyme, and it actually is an antimicrobial. It causes the uh, basically it just causes the cells to fall apart. We also have it in our saliva. But just blinking helps flush out dust and pathogens, microbes, I should say. So saliva is constantly flowing in your mouth. So we're always swallowing all day long, and every time we swallow these pathogens, and the acid in your stomach kills them most of the time, not all of them. Also, when you get an infection, a lot of times your body will sense the chemicals that are produced by the pathogen, and it will cause sort of an evacuation, a flushing of whatever's in your GI tract. So you'll get vomiting and diarrhea, and that's just an effort to just get rid of whatever happens to be inside there. And the most common type of an infection is an infection with the portal of entry through your mouth. So a lot of times you're going to get diarrhea and nausea and vomiting, even if it's not necessarily that type of an infection. It's just a reaction that our bodies have. And a lot of people even get more sensitive to it, where you little things will begin to trigger nausea and vomiting. Um, and you just get more and more sensitive to it over time for some reason. So in the respiratory tract, these are also, remember, mucous membranes, but one thing we have in the respiratory tract that we don't have in like the genitourinary tract, in fact, the only places oh, I'm getting ahead of myself. Uh, so in the what are called the vestibules, which are just the nostrils, the kind of opening in the nostrils, we have hair, which are called vibrissae, which I think comes from the root word of where we get the word bristle, because they're these short, stiff hairs. Of course, as you get older, like I am, they start getting longer, which is a little bit annoying. Anyway, they kind of help trap dust and pathogens right at the entrance. They don't do a great job, but it's like a rough filter. Um, Also, we have a flow of mucus because these are mucous membranes, and that helps to flush it out. When your nose begins to run, that's kind of a double-edged sword because, and I don't think people really know, if that's if it's a reaction by your body on purpose to get rid of the pathogen, or if it's an adaptation by the pathogen to cause your nose to run so that it can spread itself. It probably does both, and who knows which, reason it began as, maybe the chicken or the egg kind of a thing. Coughing sort of and sneezing, those sort of fall into that same category. Is it to get rid of the pathogen or is it to cause the pathogen to spread? I don't know, probably both. And that's the next one. So anyway, those things just get rid of the pathogen. Okay, so this is what I was going to talk about. So also inside of the respiratory tract, we have these things called cilia. So ciliated epithelium is a type of epithelium that has cilia on the surface. Now, cilia, this isn't a great picture um, because, if you need to review this, go back to one of your anatomy books and look at it. But cilia are these projections that go through the cell membrane, which are not to be confused with microvilli, which are what these things are right here. At least that's what it's trying to show. Um, so these longer ones right here are the cilia. Now, you have microvilli inside of your um, GI tract because we use it to absorb. And they make these things look like short little bumps, but in your GI tract, they're quite long and deep. Anyway, microvilli don't move, cilia do. Okay, so these kind of will look like seaweed in the ocean floor. Only difference being cilia are what's doing the moving, where on the ocean the seaweed is being moved by the ocean. So it's kind of the opposite, but they sort of look similar to that. And what they do is they sweep the pathogens and the mucus that they're trapped in across the surface. Uh, the example I use is crowd surfing. So if you grew up when I did, when crowd surfing was invented, you'd jump on the crowd at a rock concert or whatever, and then you go, everybody's hands push you across the top of them, and hopefully you don't wind up in the mosh pit where you're going to get beat up. Anyway, that's sort of an analogy of how this sort of works. So the three places that I know of that we have cilia are in your respiratory tract. Females have them in the fallopian tubes, and then we also have them in the ventricles of our brains. And they are not always for the same purpose. It's always for moving something, but in this case, we're talking about these physical barriers. In the genitourinary tract, we constantly have a continuous trickle of urine, not through the urethra, thank goodness, but through the ureters, which is from the kidney to the urinary bladder. So it is always going downhill, right? At least if you're standing up in anatomical position. And so it's constantly flushing um, out of the kidneys down into the urinary bladder. And then, of course, we periodically empty the bladder, which flushes out the urethra and helps to get any kind of pathogens that might be sticking to the urethral walls out. And don't forget, we talked in a previous chapter about normal flora. Those are also in these areas, in some of these areas at least, especially the genitourinary tract, and they also produce competitive secretions like acids. So we also, not we, but women have vaginal secretions that also help to flush out these pathogens in their reproductive tract. And I always call it the genitourinary tract. Not everybody does, I guess, because they're so closely related. But I guess it's important to make note. In men, it's the same tract. In women, however, it's not because you have a urethra. I shouldn't say you because there might be men listening to this. Because women have a urethra, but they also have a vagina, which are separate. All right. So anyway, there's two different areas we're talking about with women. So that brings us then to chemical barriers. So these are non-specific, meaning they're just there and they just um, compete with whatever they happen to be effective against. So sebaceous secretions, which are from the sebaceous glands on your skin, remember the secretions are called sebum. So it's an oily liquid that is secreted. More so like on your face. That's why your face gets greasy. But really all over, everywhere pretty much besides the palms of your hands and the soles of your feet. I'm pretty sure there's not any sebaceous glands on those two areas. But the secretions from the sebaceous glands just make it difficult for colonization of bacteria and fungi. Um, so in your eyelids, there are glands. There are also tears. I'm not sure if that's if they're including this or if it's coming up. Um, but they also produce substances that make it difficult for the bacteria to colonize. So inside our tears, apparently I was ahead of myself on this, but lysozyme, which don't forget is also in the lysosomes of pretty much all of your cells, um, aside from probably your red blood cells and cell fragments like your platelets and things like that. But pretty much all of your cells, we have lysosomes with lysozyme in them. So it's the same enzyme, it's just for a different purpose. So as I said, they just destroy the pathogen by hydrolyzing the peptidoglycan layer of the cell wall of the bacteria. So in your sweat, I'd mentioned the acidity of it. There's lactic acid that is secreted in your sweat and a high electrolyte content, which is essentially, well, I shouldn't say it's salt, but it is definitely salts in it. Sodium, potassium, things like that, as well as chloride ions. And those things will cause desiccation of the bacteria. It's the reason why we create or why we have ham and things like that, to be able to preserve meat because the bacteria can't live in that high osmotic environment. So the acid obviously makes an acidic pH, which is a low pH. But there are also fatty acids from the sebum that evidently, I don't know how exactly, but they somehow compete with the bacteria or make it so the bacteria uh, can't thrive. Another chemical barrier, we had already mentioned this, but the hydrochloric acid, HCl, hydrogen chloride, in the stomach, it kills bacteria. It's a super, super low pH, like 1 to 2 pH. And so when the bacteria are exposed to it, it kills most of them, not all of them. Certainly the vegetative bacteria, but endospores and things like that could potentially survive. So the same digestive uh, enzymes and whatnot that we produce in the small intestine, and the liver as well, bile, which is produced in the liver. Also, because they destroy food, I mean, that's the reason why we produce them, is to break the food down. They're proteases and lipases and um, enzymes and chemicals that break up uh, sugars and pretty much everything, so carbohydrates and whatnot. Well, the bacteria are also made of those same organic molecules, and so they will get destroyed by the digestive juices as well. So males produce semen, which is the mixture of sperm and the secretions from the seminal vesicles, the bulbourethral glands, the prostate gland, um, Cowper's gland. Anyway, they also create um, certain chemicals that are antimicrobial. I had mentioned this earlier, and also in other chapters, that the acidic environment of the vagina also defends against bacteria. And there's also acidity in the male urethra as well because of the normal flora. Okay, so genetics was the other one that we had mentioned. And we're not necessarily at risk of being infected with every single pathogen that's out there because our genetic makeup isn't necessarily compatible. However, there's this constant evolutionary flux of both host and pathogen that could potentially make us either more susceptible or less susceptible um, just as time goes on. So the first line of defense is not enough to keep us from getting infected. And because of that, we have also a second line and a third line of defense. The second line of defense are those things listed there, which we're going to get into next. For each of the barriers below, state whether it is a physical, chemical, or genetic barrier. Hydrochloric acid of the stomach is obviously a chemical barrier. Sloughing of the skin, or just the skin itself, is a physical barrier. Lysozyme in the saliva and tears, obviously a chemical barrier. Mutation in the genes for complement proteins would be a genetic, but of course the complement proteins are chemical. Uh, the ciliary escalator, in other words, the um, ciliated epithelium that pushes things up, at least up your esophagus and your bronchial tree. Uh, we also have cilia inside of the sinuses, which don't necessarily go up. But anyway, that would be, of course, a physical barrier. All right, so let's overview the second and third lines of defense again. So there's this word called immunology, which if you break that down, obviously is the study of, immunity, which would be the second and third lines of defense. We're only going to discuss the second line pretty much in this. We'll go a little bit over the third. We'll go into detail of the third line of defense in the next chapter. But a healthy, functioning immune system is responsible for surveillance of the body, which is just going around and just, looking for the bad guys. It's like a policeman who drives around looking for crime, right? Recognition of foreign material, which could be living or non-living, and destruction of those things that are foreign. So white blood cells are constantly moving throughout the body, both in the blood as well as in the tissues, because we have all kinds of resident white blood cells. So they have the ability, when functioning properly at least, to recognize the body's own cells, which we call self, as well as non-self, foreign things. And the way they do this, they don't walk around looking for people they recognize or cells. It's all by chemical reaction. So when they come next to a cell, there are antigens and antibodies, or I guess in this case there are antigens and receptors on the white blood cell. And just like this picture is showing over here, this purple cell right here is the white blood cell, and these are the receptors on it, and they match these normal blue receptors or antigens here, which are the cell markers. And so if they match, then there's no reaction that occurs. The white blood cell leaves this cell alone, whatever it happens to be. Down here, though, these what are called PAMPs, which are also antigens, cell markers, but these are called pathogen-associated molecular patterns, which they're just foreign macromolecules, basically. They don't match. So when the pathogen is recognized by the white blood cell, like this one obviously is a macrophage, then it phagocytizes it and destroys it, and then we're going to get to what happens afterwards in the next chapter. Now, a problem that can occur regarding all of this are these things called autoimmune diseases. And auto means self. And what that means is you get immunity against yourself. In other words, you attack your own tissues, and that's just because of a breakdown of the recognition. So these chemical reactions are messed up, and when these white blood cells come into contact with certain uh, self cells, such as like rheumatoid arthritis, um, your white blood cells attack the synovial lining inside of the joints. Um, anyway, with these autoimmune diseases, we attack ourselves because we don't recognize our own cells as self. We recognize them as foreign or non-self. So as I've mentioned, the way that it does this is by recognizing these markers, or what we call antigens. And these things usually consist of proteins or sugars. They're not giant, but they're also not super, super small. It takes a certain number of atoms, a certain sized molecule, in order to trigger this most of the time. And we'll get into that a little bit later, I think, the next chapter. But anyway, these are what our body recognizes as self or non-self. So we're going to talk about some specific body compartments regarding immunity. The first one is called the reticuloendothelial system. The second one is called the extracellular fluid, or ECF, also known as interstitial fluid. This is the fluid that is surrounding our cells. Other books I have used call it tissue fluid. The bloodstream, and the lymphatic system. So some of these body compartments actually have communication between each other. And that's because we need things to be able to move in and out of them. So cells, chemicals are able to go back and forth from the blood and the lymphatics because different things occur in different areas, right? And once we produce what we need to produce in, say, a lymph node or the spleen or something like that, we need to be able to get it back into the blood so that it can do its job, which is fighting off infection. So some of these are directly through just a vessel, but others of them can actually move through the wall of the vessel. I don't know if we've talked about it, but there's this it's called diapedesis, where a white blood cell will actually go between the epithelial cells of a capillary and go into the interstitial space. So the first thing we're going to talk about of these compartments is the reticuloendothelial system. And I'm going to tell you, this is a little confusing to me because this is so closely related with the lymphatic system that I'm not entirely clear on why this particular book separates it as its own system or compartment, but it does. But just keep in mind that this is very closely related to the lymphatic system because it's through the entire lymphatic system. So reticulum is just a word that means these small, really delicate fibers. They're connective tissue, but they're weak uh, fibrous connective tissue. They're not like collagen fibers, which are very strong and very dense. So in this picture, it's these tiny little thread-like things, and this reticulum is really common in areas such as I don't know, your fat tissue. Uh, they're what provides some of the structural support in those tissues, even though it's very weak structural support. So also throughout this reticular endothelial system, we have lymph capillaries and blood capillaries, and that's what allows these cells to be transmitted back and forth across these different tissues. So the reticuloendothelial system, and remember I said that these are very closely associated with the lymphatic system, but tissues or organs that are part of this include the thymus, which is part of the lymphatic system. And this is where we have white blood cell maturation or lymphocyte maturation. And not only maturation, also production. We'll get into that later. Lymph nodes, which are obviously part of the lymphatic system. Tonsils, which are um, clusters of lymph nodules, which are obviously part of the lymphatic system. The spleen, which is part of the lymphatic system. And lymphoid tissue in the mucosa and the gut. Remember the galt? I think we talked about that. Um, but also in the respiratory tract underneath the mucous membranes or mucosa. So inside of this reticuloendothelial system, we have macrophages or macrophages. So these are white blood cells that are constantly roaming around or um, living in tissues like dendritic cells and things like that, waiting for uh, pathogens to come in so they can attack them. So in your skin, in your lungs, in the liver, those are called Kupffer cells, and in the lymph nodes and spleen, bone marrow, basically all over your entire body, as well as in the blood, uh, circulating around your whole entire systemic circulatory system. Well, your whole circulatory system. Okay, so that brings us then to the lymphatic system. So a network of vessel cells and specialized accessory organs. So remember the lymphatic system and the reticuloendothelial system go hand in hand with each other. So there's two functions of the lymphatic system. The first one is to drain the excess fluid. So you may need to review. Actually, it's not important for this class, but um, when we produce tissue fluid because of filtration in the capillaries, um, on the arterial end of the capillary, um, that fluid accumulates in the interstitial space. We call it interstitial fluid or tissue fluid. We already mentioned in this chapter, but we can't leave it there, right? And so we create, because of the loss of fluid, but leaving certain dissolved particles such as albumin inside of the blood, on the venule end of the capillary, we end up with what's called a colloid osmotic pressure gradient. And so we reabsorb on the venule side of the capillary like somewhere in the area of 80% of the fluid, but that leaves 20% of the fluid that was created by filtration still in the interstitial space. And so if we don't take care of that, it just, of course, accumulates, right? And there are diseases where that happens. It's called elephantiasis. So we've got to get rid of it. And the way we get rid of it is with the lymphatic system. So we have lymph capillaries that drain it, and it ends up getting back to the blood. So that bullet point right there is what I just said. So it's a drain-off system. It also functions for draining inflammation as well. But the other function that we're going to focus on in this chapter, and this is the important function, is the surveillance, recognition, and protection of these foreign materials, cells, those sorts of things. So lymphatic fluid, also known as lymph, is a plasma-like liquid because it was plasma right before it became lymph. Actually, that's not true. It starts out as plasma, leaks out into the interstitial space, where it's not exactly plasma because the larger proteins can't make it through. They're filtered out, and there's obviously no blood cells, which isn't part of plasma. That's part of blood. But anyway, it's close to plasma. And then, of course, what comes into the lymphatic system is essentially the same thing. The only difference is in the interstitial fluid, there are a lot of waste products that come out of the cells, and those are also drained back into the lymphatic system. So formed because of plasma filtrate, I've already mentioned this, diffuses into the lymphatic system, and then it transports white blood cells, fats, and that's primarily in the small intestine, and cellular debris. So we're constantly having cells dying, right? And so when the cells are destroyed, we have to get rid of the parts, and so we bring them back out through the lymphatic system. And we also use it to transport infectious agents out. So lymphatic vessels are similar to veins in their anatomical makeup. In the books, we always draw these lymphatic vessels as green. It's important that you understand they're not green. They just use that color to differentiate them from the red and blue blood vessels, veins, and arteries, which, by the way, are also not red and blue. I'm sure if you're taking this class that you already knew all that, but just to clarify. Anyway, so they often accompany blood capillaries. So where you see a capillary bed in your blood circulatory system, you will also pretty much always see a lymphatic capillary bed. So they're in all parts of your body except they are not in the central nervous system, they are not in the bone, not in the placenta, and they are not in the thymus. So we have very high numbers of them in the hands, feet, breast, and these are basically in areas where there's a high likelihood of the introduction of pathogens. Hands and feet, because it's a common place to get cuts, right? Breast, because there's an opening there, and nursing babies apparently are dirty, I guess. I don't know. So a little bit of an anatomical review. Lymph flows from the extremities. Well, this would be the upper extremities. I mean, everything flows into the subclavian vein eventually, but there's more of a direct route of the upper extremities. That's kind of a misleading thing. So yes, let me just clarify. So all of your lymphatic vessels will eventually flow into the right and left subclavian vein. We're not going to get into it in this particular chapter, but you might want to review the right upper quadrant of the body, which includes the upper extremity and part of the chest and head, all goes into the right subclavian vein, and then all of the rest, which includes both lower extremities, goes into the left subclavian vein. Not really important for this chapter, or for this book. So lymph moves through what's called the skeletal muscle pump, similar to how the veins flow in the lower extremities. There's these valves throughout the lymph vessels that it functions like a ladder. So as you squeeze the skeletal muscles, it pushes the lymph up, and then the valves close, and it doesn't allow it to drop back down. So the next structure of the lymphatic system is called the thymus. So this is the site for T cell maturation as well as T cell formation. We actually have stem cells that will actually produce T cells, but some of the T cells are produced in the bone marrow and migrate to the thymus, where the hormones in the thymus, called thymosin and thymopoietin, so we call those thymic hormones, actually mature the white blood cells and turn them into T cells. So the thymus originates in the embryo as two separate lobes embryonically, and then they fuse together to become one organ. So in the thymus, these cells are actually, quote unquote, trained how to become, sort of warriors, I guess, and actually be able to start to recognize the bad guys. So the hormones are thymosin and thymopoietin, as I had mentioned earlier. Now, once these T cells are actually produced, they will then migrate to other lymphatic tissues, such as the lymph nodes and the spleen, where they will then stay and wait for these things called antigen presenting cells to come and bring them antigens from bad guys so that they can produce antibodies and other specific immunity. Okay, lymph nodes are small and they are encapsulated in connective tissue, these little sort of bean-shaped organs. There's different sizes of them, but something around the size of a lentil, if you know what that is, usually they can be enlarged, especially following or during an infection. So they're found along these lymphatic vessel pathways, and they're clustered in specific areas, such as in the inguinal area called inguinal nodes, um, axillary nodes, in the axilla, and so on. And the reason why they're in those areas, I have kind of mentioned that earlier, is because these are very common routes of infection. So if you've ever noticed what people call swollen glands, around your neck, those aren't glands, those are lymph nodes, because the most common portal of entry for infection is through the face, through your eyes, nose, and your mouth. And so when you get a head cold, those lymph nodes are fighting off infection, and so they get sore and swollen. But as I had mentioned earlier, the hands and the feet are other common pathways. So that's where you'll see a large clusters of these lymph nodes. So when you go to the physician, they feel your neck, and that's one of the things they're feeling for is swelling of these nodes, and that just gives them a better idea of what is going on with the infection. Okay, now lymph nodules are different. These things are very, very small. Small as like 1 millimeter. They're unencapsulated, so there's no connective tissue around them. They're still along these small vessels, and they have a similar function, but we see them a lot more around the respiratory and the GI tracts. Your tonsils are also collections of these lymph nodules. But aside from that, they actually have a fairly similar function to the lymph nodes regarding immunity, at least. The spleen, found in the left upper quadrant of your abdominal cavity, is similar in function to these other tissues that we've been talking about, the lymph nodes and the lymph nodules, but there is one massive difference, and it's that it filters blood where the others filter lymph. Okay, so flowing through the nodes and the nodules is lymph. Flowing through the spleen is blood. So if you remember the hepatic portal system, you know that the spleen drains into the hepatic portal vein, which drains into the liver. And you'll need to go and review why. I don't think we talk about it in here. but it's primarily because of the other function of the spleen is to um, recycle red blood cells that have worn out. So we call this the reticuloendothelial system, which we've already discussed. But that reticuloendothelial system has a huge function in um, recycling these red blood cells. But the other thing that reticuloendothelial system does is filters out the pathogens so that the macrophages can phagocytize them. So adults that don't have a spleen it's fairly normal. If you have enough damage to the spleen, they don't even try to repair it. They just take it out because it doesn't really affect your life much. There is some increase in certain um, diseases for some reason. I'm not entirely sure why. Um, but aside from that, you function fairly normally. Now, children, on the other hand, have some issues. Now, really, really young children and fetuses won't survive. They have to have a spleen because they don't have developed red bone marrow, and so they don't produce red blood cells. And so we use the spleen when we're very young to produce red blood cells, but once we have red bone marrow, we don't need it for that anymore. However, they still use it to produce white blood cells as well, and so they are immunocompromised. Another function is for the storehouse of blood. So your spleen can release massive amounts of blood in the case of a hemorrhage. And another thing that for some reason isn't mentioned here is it's also a storehouse for platelets as well. Okay, so miscellaneous lymphoid tissue that's not talked about already. One of them is called gut-associated lymphoid tissue, also known as GALT. And these are these bundles of lymph nodules under the intestinal mucosa. They serve a couple of functions. One is for immunity, but the other one is just to help to drain the fatty acids that we from the food that we eat. But it is a competent first strike or first line of defense against the influx of microbes that are in the food that we eat. Our food is pretty clean nowadays. Um, it used to be pretty much filthy. However, it's still, even the food that we eat today still it's not sterile by any stretch of the imagination. So there's still pathogens in the food that we eat. I just said to first line of defense. I shouldn't have said that because that's a specific term that we use. So let's just use what's written here in this bullet point, first strike. So not first line of defense because this is part of the second line of defense, right? Another structure is called the appendix. Everybody knows what that is. The significance of the appendix isn't really agreed upon, so we'll not talk too much about it. These things called lacteals. These are the things that are in the villi of the small intestine and just help to drain the villi for both um, uh, draining pathogens, but also draining um, macromolecules that were produced from the breakdown of our food. Now, also along the small intestine, we have these things called Peyer's patches. That's capital P, named after whoever named that, Mr. or Miss Peyer, I guess. Aggregations of lymph nodules in the small intestine, primarily in the ileum. And the function is the same as what we've been talking about. Okay, so similar to the GALT, we have, which is gut-associated lymphoid tissue, we have this lymphoid tissue in the respiratory tract, and I've mentioned the tonsils as well. And when women are lactating or nursing, they are also producing um, antibodies in the lymphoid tissue in the breasts. And these don't function all the time, evidently. Um, but as I mentioned earlier, it's a really common place to have pathogens introduced during nursing, not only because there's the fact that there's holes in the nipples where the mammary glands secrete, but also because it's very common for um, cracks and fissures to occur on the nipple because of the suckling of the infant. All right, so this figure in your book just kind of gives an overview of the different circulatory systems, right? So this is what we call the circulatory system, which is blood and includes the veins and arteries. This obviously is the lymphatic system. And then this right here, let's see, it's just trying to show, I guess, two things. One, that the circulatory system goes in a circle, so it goes around and around and around, where the lymphatic system doesn't. Because these start out as what we call blind capillaries, which means they just start. They aren't fed by another vessel, where our capillaries in the circulatory system are fed by the arterioles, right? Anyway, so they just go up, and then it shows that they empty back into the venous blood, which is the subclavian veins on the right and the left. So let's talk about blood for a minute. This is going to be in regards to immunity or the immune system. There's certainly a lot more to talk about besides what we're going to talk about here. But whole blood. Whole blood consists of blood cells, which are called formed elements, as well as the plasma, which is the liquid part of the blood, and what is called the serum, which is the same thing as plasma without the structures that form the clots. And so we use the serum for immune testing and therapy. So if we're checking to see if somebody has an antibody titer for, I don't know, the flu or something, we'll use serum to do that. So hematopoiesis is a fancy word for production of blood cells. Hemat means blood, poiesis means formation. Stem cells we have lots and lots of stem cells throughout our body, of course, for all kinds of different cells, because all of our cells are produced from some sort of a stem cell, but this is in reference to blood stem cells. So our stem cells for blood are, as an adult, primarily in the bone marrow, right? We did talk about the spleen as well, but for this conversation, we don't need to really discuss that much. So as these stem cells divide, they will differentiate into specific types of white blood cells. And so what a stem cell is sort of a blank cell that kind of can become anything. Not anything, but any type of a blood cell. So we can either get red blood cells, white blood cells, or platelets, which come from something called a megakaryocyte, which is a type of a cell, because platelets themselves aren't actually cells, they're just fragments of cells. So white blood cells, also known as leukocytes, can develop into one of several types of white blood cells, which include lymphocytes, and primarily responsible for the immune function. So this figure in your book shows the 7 types of white blood cells and how they all differentiate from one stem cell. So let's kind of break this down a little bit. So here's the stem cell up here, and it has the choice to either become a stem cell for one of these blood cells or a lymphoid stem cell, which will become eventually a lymphocyte. So they differentiate into a lymphoblast, and then which will either become a T or a B cell. So over on this side, though, these are all of the blood cells. So if this hematopoietic stem cell does not become a lymphoid stem cell, it's going to become one of these other types of blood cells. So either an erythroblast, a megakaryoblast, a myeloblast, or a monoblast. So an erythroblast, as you can see, will become a red blood cell. A megakaryoblast will become a megakaryocyte, which will then fragment and become platelets. Over on this side, if it becomes a myeloblast, and these blasts are all precursors to these other types of cells, obviously. So a myeloblast can differentiate into one of these 3 types of white blood cells called either neutrophils, basophils, or eosinophils, and these are all called granulocytes. And the reason why they're called granulocytes is because they contain these inclusions called granules inside of them. Now we're not going to spend a whole lot of time talking about basophils. or eosinophils. We will talk about neutrophils some a little bit later. But one more thing we need to mention about these myeloblasts is not only can they become one of the types of granulocytes, they can also differentiate into a megakaryoblast, which will eventually become a platelet. Or, as you can see this line right here, they can become mast cells, which, along with the basophils, are responsible for a lot of the inflammatory response. Okay, so going back up here then, the last one we haven't talked about here on these blood cells is this monoblast here. So these are the 7 types of white blood cells here, mast cells, and then these 6 granulocytes and agranulocytes. So the monoblast will differentiate into a monocyte, which will become macrophages or dendritic cells. So dendritic cells are essentially macrophages that don't circulate in the blood. But both of these types of cells here are phagocytic cells and come from monocytes. Now, last thing, these lymphoid stem cells, they will differentiate into what are called lymphoblasts, or directly into what's called a natural killer cell. And there's actually another type of natural killer cell we'll talk about later, but right now we don't need to worry about that. But either a T or a B cell, which we'll talk about why those are named that way later, and what they do later. So we don't even need to talk about this right now. But that basically goes over the basics of how we get our blood cells from a stem cell. So take some time to go over that on your own in your book. Lymphatic fluid contains all of the following except white blood cells, extracellular fluid, red blood cells, cellular debris, or infectious agents. And the one thing that at least it should not contain is red blood cells, because red blood cells are filtered out. In other words, they stay in the blood when the filtration occurs to produce the extracellular fluid. Now, of course, you could argue that it doesn't contain extracellular fluid because as soon as it leaves the interstitial space and goes into the lymphatic vessel, it's no longer called extracellular fluid, it's now called lymph. But, you know, we don't need to get quite that technical. Okay, let's get more specific about this second line of defense. And this is what we'll spend the rest of our time for this chapter on. So these are generalized and non-specific defenses that support and interact with this specific immune responses, which is the third line of defense. Now, the first line of defense was also generalized and non-specific, but those were barriers. Okay, so the second line of defense is not barriers. These are cells, right? So the main mechanism that the second line of defense uses are mechanisms of phagocytosis, inflammation, fever, and antimicrobial proteins. So let's just go down through that list and discuss all of these things. So phagocytosis, if you break down that word, phage means eat or to eat, cyte means cell, and osis just it literally means condition of, but in this case it just means you know the act of a cell eating, so to speak. So what's involved in phagocytosis? First of all, surveillance. So they have to survey the tissues and look for the bad guys, right? Or also injured or dead cells that are our own cells as well. They get marked for phagocytosis as well. They then ingest and destroy those tissues. They also extract the information, which would be the identifying markers of the bad guys. and then they will present them to the third line of defense that we'll get to next chapter. So, professional phagocytes, meaning this is what they do, are neutrophils, monocytes, macrophages, which come from monocytes, right? And also include, well, I shouldn't say they include, but another type of monocyte would be like a dendritic cell, right? So neutrophils then. These are general purpose phagocytes. These are very responsive in the early infections and early inflammatory responses. So a high neutrophil count, which is called neutrophilia, which you would detect from doing blood work, is a common sign of bacterial infection. And this is the primary component of pus, right? Pus is white because it's full of white blood cells primarily, which are mostly neutrophils. So monocytes and macrophages are essentially the same thing, and as I said, include dendritic cells, right? And one thing I should probably mention, I should have mentioned this at the beginning of the chapter, I didn't think about it. You're going to read different things in different books that are going to not necessarily agree. Don't panic about it. Things change. There's also not always agreement on how all of this stuff functions and works. So I may say something that is either just plain wrong, either because I'm wrong or because my sources are wrong, or potentially may disagree with something else that you have read in another book. It's not always easy to determine which is right and which is wrong. Oftentimes, I think we just decide that whatever we learned is right. I know that's sort of what I tend to do, although that's certainly not the case all the time. Anyway, it's just something that you're just going to have to be aware of and just kind of keep in the back of your mind, and don't get too frustrated about it. So as we said earlier, monocytes are transformed into macrophages after they migrate out of the bloodstream and into the tissues. They can be dendritic cells or Kupffer cells, which is actually what this is showing. So they call these Langerhans dendritic cells, or what we usually just call them dendritic cells. The reason why they're called dendritic cells, by the way, is because they have these little dendrites, which are branches off of them. Kupffer cells are these resident macrophages actually in the liver. So these monocytes that do end up becoming resident tissue cells. Examples of these are alveolar macrophages or lung macrophages, which are macrophages that live in the alveoli of the lungs. Kupffer cells, which we've mentioned right there on the right. Dendritic cells in the skin. And there are also macrophages that are resident in the spleen, the lymph nodes, the bone marrow, the kidney, bone, brain, and so on. So all of those we just call histiocytes. And hist means tissue. So cyte means cell. So these are tissue cells, which maybe you need to be careful not to think that they are the actual tissue cell. So like they're not the liver cell, for example. Histiocytes are a type of macrophage. Okay, so there are other cells that don't take up residency in tissues, but sort of roam. So they call these dynamic scavengers. Dynamic just means not stagnant, so they're just moving. And these are the ones that will become antigen presenting cells, where they will bring them into a lymph node and present them to the B and T lymphocytes. So let's talk about how the phagocyte actually goes about destroying a pathogen. So it literally means eating cell. Phagocytosis is the act of doing the eating, right? So it's the attack and dismantling of the foreign cells. And this phagocytosis can happen just one cell all by itself as it encounters, a pathogen, or it can be part of a greater set of events called inflammation.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
Okay, so how does this all occur? The first part is called chemotaxis, and I'm going to go over all of these with some pictures in a minute. Ingestion, the formation of the phagolysosome, destruction of the pathogen, and then excretion. So there are these things called PAMPs. I think I'd mentioned these earlier, pathogen-associated molecular patterns. These are really just the markers found on the outside of the bad guys that when they come into contact with the macrophage are recognized as foreign, and that triggers the phagocytosis. So these are molecules that a lot of microorganisms have on them, but that mammals do not. So as we've mentioned earlier, they're the red flags for the innate immunity, right? The built-in immunity that you're born with. So this part does not have to be learned. So on bacteria, it's the peptidoglycan and the lipopolysaccharide of the cell wall. Viruses, though, don't have a cell wall. And so what are recognized as the PAMP on viruses is the nuclear material. So the receptors that actually can recognize those PAMPs are called pattern recognition receptors, or PRRs. So they're found on a variety of cells, and we're going to spend most of our time discussing the phagocytes. So they bind and then, of course, recognize, because what happens is a specific chemical reaction will occur once they come into contact, and that triggers the phagocytosis. And remember, this is innate immunity, built in, born with. So we don't have to come into contact with the bad guys in order to have these PRRs. We're born with these PRRs. And so the very first time we come into contact with a PAMP, this reaction occurs, which is not the same as our third line of defense, where we actually have to come into contact before we actually can recognize it a second time. So let's break this table apart in your book and go over all of those steps that we had mentioned earlier. Okay, so chemotaxis. Chemo means chemical, that's this right here. Taxi means move, right? So this is the actual movement based on the phagocyte sensing chemicals that are being released because of inflammation. So during inflammation, we produce things like cytokines, bradykinin, leukotrienes, those sorts of things, and those chemicals will actually attract other defense cells to the area via chemotaxis. I think I've probably used this example, but it's like a hound dog sniffing out, a raccoon or whatever. Okay, so once this phagocyte is in the area, it's got to recognize it. Okay, so these little guys here are the bacteria. This huge guy right here, that's the white blood cell we're talking about, this phagocyte. Okay, so the second part of this is adhesion, and this is where the bad guy, the bacterium in this case, or three bacteria actually, attach to the PRRs. So the PAMPs attach to the PRRs of the phagocyte. Once it recognizes it as foreign, it is going to engulf it and form a phagosome. Okay, so this right here is the engulfment, and I'm not really sure why, but these little guys should be inside here. So just pretend there's what, 1, 2, 3, 4, 5, 6 little bacteria inside this little what's going to become a vacuole in just a minute. Okay. Anyway, so this pinches off, and this is the same vacuole just shown in 4 different steps. Okay. So all these, this started out as this, and then it becomes this, and then we'll go through all that. But just understand, this is all the same thing. It's just showing it 4 different times. Okay, so once the phagocyte has made contact with its prey, which is a bacteria, it extends these pseudopods. And this picture doesn't show it very well, but pseudopods are these extensions that come up and out. This would be one right here. Pseudopod just literally means false foot. So it's like a foot that comes out and grabs, you know, the bad guy and pulls it in and sort of creates this vacuole. Anyway, that vacuole is called a phagosome. So the vacuole, this outside part of the vacuole, used to be the cell membrane of the phagocyte. Okay, but of course then it pinches off and becomes this vacuole. So this fluid inside here was the fluid on the outside of the cell. And another thing that the phagocyte does is release cytokines, which just contributes to the inflammatory response and kind of helps create that snowball effect, which isn't always a good thing. Okay, so we have the phagosome. What happens then is we combine lysosomes with it, and that becomes a phagolysosome. Okay, so here's the phagosome. Here's the lysosome, which contains, of course, lysozymes, right? They combine together here in step 5, and the lysozyme, the membrane of the lysozyme then just fuses and becomes part of the membrane of this, what is now called phagolysosome. It releases these lysozymes in there, and that begins to go to the next step, which is to destroy the bacteria that are inside of the phagolysosome. And this next part comes back to what we had talked about in previous chapters of how what types of chemicals we use to destroy pathogens. So we have the oxygen-dependent system, which includes the respiratory burst or oxidative burst, because oxygen-containing molecules and forms of oxygen are very, very destructive, right? Another one is myeloperoxidase, which is an enzyme in granulocytes. And they form halogen ions, which are really oxidizing. So they will actually begin to react chemically with parts of these bacteria and tear them apart. Another one is hydrogen peroxide, and then superoxide anion. And then that singlet oxygen that we talked about in previous chapter, as well as hydroxyl free radical, which is different from the hydroxyl ion, which is also very destructive, by the way. And the other system includes things like liberation of lactic acid or lysozymes, which we already talked about, nitric oxide, which somehow kills bacteria. I don't know how, but it also inhibits viral replication because of it disrupts the chemical processes that are necessary for viral replication to occur. There are also cationic proteins, which are positively charged polyatomic ions, and they act as proteolytic or hydrolytic enzymes that damage the cell and destroy it. Okay, so once we've destroyed it, we don't want to leave it in there, right? So that is where elimination comes in, and this is where we release the pieces of it via exocytosis. Okay, some of it's just released out into the interstitial fluid or the blood or wherever this phagocyte happens to be. Some of these, though, are then taken to a lymphocyte and presented to one of the lymphocytes. And that's phagocytosis. So let's talk about the inflammatory response.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries.
Section as well.
Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
There was so much in there. We could probably spend days just talking about that 1 minute video. Maybe some things to think about. So you heard me mention, hopefully, if you understood what I said, that this is showing a picture of a splinter. And then with the splinter, you can see that these, I guess these are bacteria. I may have missed that, but usually there's going to be bacteria on the splinter, and so they're going to need to come in here and combat that. Some stuff to think about. Inflammation is a local response, and so it's initiated by the tissues where the problem occurs. Another thing to maybe think about, remember these mast cells over here that it showed, these are granulocytes. And remember I said granulocytes are things, are cells that have something in them, these inclusions in them. Well, one of these inclusions were these, was this histamine. Now, histamine creates an increase in the permeability of the blood vessel, of the capillary. Okay, so in other words, it loosens up the connections between the epithelial cells. So there's an epithelial cell, there's one, there's one, there's one. And so it creates these gaps. The word I use for the neutrophils leaving, migrating out of the capillary is called diapedesis. This video said extravasation or something. I'd never heard that word before. I assume it's the same thing. If it's not, then that's news to me. Again, you're going to see different things in different books. And I don't think your book even uses that term. But anyway, so once all of that sort of occurs, you end up with phagocytosis by these neutrophils of the bad guys, right? So don't forget, all of this is chemical reactions. I've said it before, but our bodies function like a computer, right? If A happens, then we do B. If C happens, then we do D, right? All of it is chemical reactions that occur. There's nothing intelligent about it. And that's not to say it's not complicated. It certainly is. But there's nothing mysterious about it. We just don't understand it all. It's just not magic, I guess, is the point I'm trying to make. Okay. So the inflammatory response is diagnosed with some specific signs. Rubor, which is Latin for red, redness. And this is caused by the increase in circulation and vasodilation. So and that's primarily because of histamine, by the way. It causes vasodilation and an increase in the permeability of the capillaries. And by the way, that increase in the permeability, if you think back to that picture that we were just looking at on the video, that not only does that allow neutrophils to come out, it also allows fluid to come out at a much higher rate. And that's what creates fluid accumulation in the tissues, which we call edema or swelling. Calor, which is just a word for heat or warmth. It's where calorie comes from. And that's because when you increase the blood flow to an area, you increase the heat because the blood comes from the core, which is warm, right? Tumor or tumor, which is swelling. That's what that word literally means. And again, this is because of the fluid that escapes. And then dolor, which means pain. The Spanish word is duele, or dolor. It actually is dolor, actually. And pain comes from sensitization of the free nerve endings in the area, which makes them fire when they normally would not fire. And that is so that we become aware that there is an issue so that we can consciously take care of it. At least that's the theory. Okay, so there's a lot of evidence. I've been hearing about this for over 10 years now, but it's actually becoming more mainstream than what it was when I first began hearing about it. And I don't know how long people have been theorizing this, but there's a ton of evidence, and there's becoming more and more all the time, that a lot of chronic diseases, and some can almost argue almost every chronic disease at least has a component, if not a large portion of the cause is because of chronic inflammation. And so you get, now I said the inflammation is local, and it usually is, but it can go haywire and become systemic. And that's where we end up with these chronic diseases such as cardiovascular disease, vascular diseases such as atherosclerosis, and those sorts of things that can lead to clot formation and stroke and all that sort of stuff. A lot of people think that aging is a consequence of inflammation. Certainly there's a component of oxidation from free radical damage, but probably also I'm fairly convinced that inflammation has a massive role in chronic diseases. So what sorts of things can cause inflammation to occur? Trauma from an infection, which is because the pathogens create tissue damage, right? Tissue damage or tissue death because of physical damage, such as an injury, or chemical agents, such as some sort of a chemical like an acid or something that you would come into contact with that would create tissue damage, or specific immune reactions, which we're going to get to in the next chapter. So the primary reasons we have inflammation are to mobilize and attract the immune components to the injury site so that we can fight it off or repair it. So to repair the tissue damage by the actual repair cells, which would include phagocytes, it would also include fibroblasts and those sorts of things. To destroy microbes if they're involved and block their further invasion. Now the bad thing about this inflammatory response is it can become a vicious cycle and actually cause tissue injury. Obviously that's why we were talking about the chronic diseases earlier. So some of these microbes actually have developed mechanisms to cause inflammation to occur on purpose because it benefits them somehow. So we have these inflammatory mediators I had mentioned before. So what do they do exactly? Cytokines, this is a general term for just inflammatory mediators. And so there's lots of different ones, and they do things like regulate, stimulate, suppress, or just have different ways of controlling um, cell development, inflammation, and immunity. These cytokines are produced by monocytes, by macrophages, by lymphocytes, by fibroblasts, by mast cells, by platelets, by endothelial cells, lots and lots and lots of different places. Remember, inflammation usually starts out as a local process, right? But then once these other cells get involved, they can also begin to release their own cytokines. So their effects may be local or it may be systemic. Sometimes it's short term, sometimes it's long term. They can have specific effects or they can have general effects, and they can be protective or in some cases pathologic. And this is a list of a whole bunch of these cytokines. And over on the left side here, it kind of gives the basics of what everything in this box does. And same with all the rest of these, right? And then these are some specific examples of what they do. So let's go through some of these because I think that a lot of this is pretty important. The first one is tumor necrosis factor. So it's released by a lot of different cells, and it increases the chemotaxis and phagocytosis so that these other cells can come and get involved. It also serves as, this says endogenous pyrogen. What that means is it's a built-in fever producer. In other words, it triggers the fever mechanism, which is involved with the hypothalamus, resetting the temperature control mechanism of your body temperature. Interferon, and there's different types of interferons, also produced by several types of cells, leukocytes, fibroblasts, and others. An important thing that it does, it inhibits viral replication because the virus has to replicate by cell division, so it inhibits that. It can also increase the action of some lymphocytes. Interleukin 1, actually. There are several types of interleukins, but interleukin 1, produced from dendritic cells and macrophages, similar to tumor necrosis factor up here. Interleukin 6, secreted by macrophages and T cells. The thing I want you to focus on this one is to stimulate the growth of B cells. Those are the ones that will, a form of a B cell called a plasma cell actually produces our antibodies. So we'll come back to this interleukin 6 later in the next chapter. So these were nonspecific mediators of inflammation and immunity. These are mediators that, remember, these are all cytokines, right? These activate specific immune reactions. So interferon gamma comes from T cells. It is very important in the differentiation of T and B cells into memory T cells and memory B cells and cytotoxic cells and those sorts of things. Also stimulates diapedesis, which is the movement of the cells out of the blood into this tissue, right? Interleukin 5 activates eosinophils and B cells. Interleukin 10 inhibits macrophages. Interleukin 12 activates T cells and killer cells. Vasoactive mediators, we've already mentioned histamine, and we've already talked about what that does. Serotonin, which is also a neurotransmitter in your brain, or in your, in your brain. Anyway, serotonin is produced by platelets and interstitial cells and causes smooth muscle contraction, which is in the blood vessels, right? Bradykinin also does a similar action to histamine, which is vascular permeability increase. But I think the thing that's important about bradykinin is to understand that it increases pain. And that is, what makes us aware of a problem. Okay, interleukin 2 is going to become very important in the third line of defense. So it's produced from T and B cells, and it causes both of those cells to differentiate into other types of T and B cells. Macrophage colony stimulating factor just causes macrophages to develop. prostaglandins, and there are tons and tons and tons of different types of prostaglandins, um, some of which, in fact, not very many of which, primarily prostaglandin E2 is important in inflammation. Some of them are actually anti-inflammatory. So understand there's a whole bunch of different types of these, but they cause inflammation and also contribute to pain. Leukotrienes stimulate the contraction of smooth muscle and also increase vascular permeability. And the last one, platelet activating factor, obviously causes the aggregation of platelets, which is what contributes to clotting. So this table here is essentially what we talked about, or what the video talked about. So I don't think there's any much of a reason to go over it. You can certainly look over it on your own in your book. Okay, so let's talk some more about diapedesis and chemotaxis. So diapedesis, as I said, is the migration of white blood cells out of the blood vessels. In the video, it showed neutrophils. And what gives these white blood cells the ability to do this is because they can actually move and change their shape. It's said that they move in an amoeboid fashion, which just means similar to how an amoeba moves, which you remember from chapter 1. And just like the video showed, the endothelial cells of the capillaries have receptors on them, as do the white blood cells. And as those chemotactic chemicals are released, it causes the receptors to become sticky. And that stops the blood cells and then allows them to squeeze between the endothelial cells. So chemotaxis is the response to some specific stimulus, which is obviously a chemical. And that's what causes these cells to congregate. So both of these are critical for the white blood cells to be able to fight off the infection. And this just shows a picture of really just what the video showed. And this is an actual live slide, not live slide, but an actual slide of real white blood cells. So why is it good? Well, obviously we just talked about it, so that the cells can get out of the blood into the area where the infection is. But also apparently it dilutes the toxins that are created by the infection. And fibrin, which is a protein, traps microbes so that they can't continue to spread. It walls them off, so to speak. Pus. Everybody knows what it is, but everybody knows what it is, but not necessarily what it's made of. So it's essentially mostly white blood cells, but also all of the junk that is part of the infection, the dead cells and the parts of dead cells and bacteria and whatnot. So pyogenic bacteria, examples of those are strep, staph, gonococci, which produces gonorrhea, meningococci, which produces meningitis. All of these things can stimulate the formation of pus. So along with inflammation, a lot of times people have fever, or even aside from inflammation. And a fever is elevated body temperatures above normal. So pretty much happens with all infection to some degree. It, however, can also be associated with some allergies, cancers, and some other organic illnesses. There are also fevers of unknown origin. These are super common in children. What it is, though, is intermittent fever above 101 Fahrenheit that lasts longer than 3 weeks with no known cause. So the hypothalamus, as I had mentioned before, maintains as our thermostat, keeps our body at 37 Celsius, which is 98.5 Fahrenheit. So a low-grade fever is 100 to 101. Up to 100 is considered to be normal. Some people have normally lower body temperatures. Mine usually runs around 97. Other people's runs naturally a little bit hotter. So a good average is 98.6. But a low-grade fever, 100 to 101. High fever, 104 to 106 Fahrenheit. And so these things called pyrogens, pyro meaning fever, not to be confused with something that is pyogenic. Pyo means pus, pyro means fire, which is the word we use for fever. So pyrogens are substances that cause the thermostat to be changed. It's like you go in your house and you turn the temperature up to 75. That raises your thermostat and tells your furnace to create heat, at least in the winter, until it reaches 75, or in the summer it turns on the air conditioner until it gets down to 75. So exogenous pyrogens, exo, outside of, right, are the products of infectious agents like viruses, bacteria, and so on, but also endotoxin, blood products, vaccines. There are lots of things that can trigger fever. Endogenous pyrogens are created within the body itself. So they come from monocytes, neutrophils, or macrophages during phagocytosis. So interleukin-1 and tumor necrosis factor, we had already gone over this in the table, but as you remember, those are examples of endogenous pyrogens. So what are the benefits of fever? We tend to look at fever as being a bad thing, but it's certainly not. It's a good thing. Your body is doing it for a reason, and it is not, in my opinion, good practice to try to control the fever. There's not really any evidence that fever creates any kind of long-term damage. So generally, if your body wants to be um, warmed up, you're going to feel cold, even if you're already at an elevated temperature. So you should just assist it based on how you feel. There may be some extremes where that's not necessarily true. But why does our body do it? Well, it inhibits the multiplication of these pathogens, poliovirus, cold viruses, herpes, and other types of infections like fungal infections. It also impedes the nutrition of bacteria because somehow it reduces the availability of iron. And by increasing the heat, you increase chemical reactions, which increases metabolism, which allows our body to just put everything into high gear to fight off the infection. So speeds up blood formation, phagocytosis, and some immune reactions. So in the cases where you do need to treat the fever, you first have to decide whether or not, I guess it's a good thing. But if you determine that it needs to be treated, it is treatable. Slight to moderate fever, as I said, generally you should not mess with them. Side effects can be things like tachycardia, tachypnea, which is increased breathing. So tachycardia is increased heart rate, lowering of the seizure threshold. But again, there's not any evidence, at least that I'm aware of, that febrile seizures, that's the seizures that you have from a fever, cause any kind of long-term damage. Although they're very rare in an adult anyway. What's more of an issue is if you have a bad enough infection and a bad enough fever to have a febrile seizure, it's probably not the seizure or the fever that you're concerned about. It's that you're probably extremely sick and you need to deal with whatever's causing the illness. So it's more of an indication of a worse problem. So most medical doctors agree that high and prolonged fevers, especially in people with cardiovascular diseases or people who already have seizures or respiratory issues, should be treated immediately. But if you're healthy, generally it's not as big of an issue. Okay, so we are going to talk about antimicrobial proteins. starting with a protein called interferon. We've mentioned it earlier, but let's talk about it in a little bit more detail. So interferon is a small protein produced naturally by certain white blood cells and tissue cells. So originally we thought, well, not we, but someone thought it was supposed to be, or that it was only because of the control of viruses. So in other words, just protective against viruses. But now we know that it's involved in lots of different pathogen defense reactions, as well as immune regulation and communication among the cells. So there's three major types. One is called interferon alpha, IFN with an alpha, as well as interferon beta. So it's got a beta on the other end of it. These things are produced in lymphocytes, fibroblasts, and macrophages and stimulate phagocytosis. Interferon gamma is produced by several cells, including T cells. And interferon gamma, which we'll talk about next chapter, is also an immune regulator of the macrophages, but also T and B lymphocytes. And it does have some sort of anti-tumor properties as well. And the way it does that, it alters the transcription in the genes, and it leads to all kinds of different effects within the cell, including increasing natural killer cells, increases antigen presentation, lysosomal activity in macrophages, increases nitric oxide synthase, and a whole bunch of other things. All right, so what are some other things that interferon does? It binds to the cell surfaces and induces changes in genetic expression. I had mentioned that earlier. It actually causes certain proteins to be produced and so on. But the exact results depends on which cell is being induced and so on. So all three interferons, alpha, beta, and gamma, can inhibit the expression of cancer genes, and therefore they suppress tumors, because that's what tumors are, cancer. So characteristics of them. Interferon causes the binding of viruses and other microbes to receptors in the host cell and signals the cells to produce the interferon. So they are then secreted into the extracellular fluid and cause them to bind to other host cells, which then causes them to do the same thing, so it becomes a chain reaction. And in a viral infected cell, it causes the cell to not undergo mitosis, which will keep it from replicating the virus. It also causes degradation of the viral RNA, which keeps it from obviously translating the viral proteins, because the viral RNA, the instructions are messed up. So anyway, it's very, very important in viruses, and that's why they originally thought that was the only thing it was used for. So this right here just shows how it does affect a virus. So here's the virus, and when the virus infects the host cell, it releases its RNA, where it's then replicated, and then the parts of the new viruses are also made, and then the new virus is released, right? Well, at the same time, the interferon producing genes are activated inside the nucleus of the host cell, and therefore it starts to produce interferon, which is then released. So what that does is it attaches to the receptors on a nearby cell, and that causes the other cell to produce these antiviral proteins, and that keeps the second cell from then producing new viruses like the first cell did. Okay, another antimicrobial protein besides interferon is this one called complement. I shouldn't say this one because there's tons of them. So named because it complements the immune reaction. In other words, it just assists it. So it's 26 different proteins that work together to destroy bacteria and viruses. So it's produced in what's called a cascade reaction, which there's lots of different reactions you've probably learned about that are cascade reactions. They're just sequential reactions where to produce something you have multiple reactions that have to occur in a row in a specific order. So the first substance activates the next, which activates the next, and so on. So there are three different pathways, but they all end up producing complement. And they're named according to the order in which they were discovered. So there's 4 overall stages, though, in this complement cascade. The first one is called initiation. So when C1, which is one of the complement proteins, I'll show you a picture of that a little bit later, but you can cheat and look at figure 1212 in your book. C1 is one of the complement proteins that binds to a foreign cell. And that leads to step 2, which is called amplification and cascade. So in this, C1 causes C5 to be produced and causes it to break apart from this complex of C4, C2, and C3 and bind to the membrane of the bacterium. That leads to polymerization, which is where we polymerize a bunch of C9s as well as C8, C6s, and C7s. But primarily we're going to end up with just C9s once this is all done, polymerizing and creating a chain which also binds to the surface of the bacterial cell. That is called membrane attack. In fact, what this thing forms is a membrane attack complex. And essentially it just creates holes in the membrane and causes the cell to be destroyed.
Regardless of whether the complement system is activated by the classical, alternative, or lectin pathway, the activated complement proteins are involved in several important processes associated with host defenses, including inflammation, attraction of neutrophils, opsonization, and cell lysis. Complement C3a participates in inflammation by inducing changes that cause the walls of local blood vessels to become more permeable. This facilitates movement of phagocytic cells from the circulation into the tissues. Complement 5a induces a directed chemotactic migration of phagocytes to the site of complement activation. Complement C3b functions as an opsonin. It binds to microbial cells, making them more susceptible to phagocytosis. Complement proteins C5b, C6, C7, C8, and C9 react together to form a hole in the cell membrane. When C5 convertase converts C5 into C5a and C5b, the C5b binds to the cell surface and combines with C6. C5b6 binds C7, enabling it to insert into the membrane. Binding of C8 is followed by binding of several C9s. The hole generated by formation of this membrane attack complex causes the cell to lyse.
So if that video looked semi-familiar, it's because it was almost the same as when we were talking about the histamine. Because remember, there are multiple chemicals that cause the permeability to be increased in the capillaries. So and they don't ever work all by themselves.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2a and C2b and C4 into C4A and C4B. C2B and C4B combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3B. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3A and C3b are constantly being formed from C3, but without activation they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3A and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5A and C5B. C5A enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Okay, so we're going to spend time talking about opsonization next chapter. But this right here is the classical pathway. This is the one that I'd already mentioned. So let's just go through it graphically. So remember, initiation is where C1 binds to the antigens on the, well, actually the antibodies that are bound to the antigens on the cell surface of the pathogen. That leads to step 2, which is the amplification and cascade, which is where C5 convertase is formed from these C4-2, C2 or C4b, C2b, and C3bs. That then splits up C5 into A and B, and that is where polymerization occurs. And so this is still bound to the membrane of the bacterium, right? So that's where polymerization occurs, where we then involve C9, C8, C6, and C7 and polymerize C9 to form the membrane attack complex, or the MAC, and that causes lysis of the cell. So that's the classical pathway. It's initiated either by foreign cell membrane of a parasite or a surface antibody. The alternative pathway is triggered when parts of the classical pathway are able to bind to the pathogen membranes. So it's quicker than the classical pathway and does not actually require antibodies, where the classical pathway had to actually bind to the antibodies that are bound to the cell surface. The lectin-mediated pathway involves these things called mannose binding proteins, or lectins, and they bind to mannose on the surface of the pathogen, and that's how they proceed, and how they proceed chemically is similar to the others. But mannose is a sugar. It's a monomer of aldohexose type of sugar, and they just so happen to be on certain pathogens, and we use them in our own body as well. That's one of the ways we glycosylate proteins. So that was complement. So iron binding proteins is another group of proteins. So humans as well as bacteria need iron in order for our enzymes to function properly. If you remember from one of the previous chapters, we use those things as cofactors, right? So iron can be a rate limiting factor in bacterial growth. In other words, you can control the growth of the bacteria by limiting the amount of iron available. And what iron-binding proteins do is they bind to the iron and make it so that the bacteria have no way of using them. So hemoglobin is actually an iron-binding protein. We use iron in the hemoglobin. That's what the oxygen attaches to. Another one is called transferrin. It's found in blood and tissue fluids. Another one is called lactoferrin. And that's transferrin, not transferrin, because fer is iron. Anyway, lactoferrin, transferrin. Lactoferrin is found in milk and saliva. That's where it gets the name lact from. Ferritin is found all over in all of our cells. And as a defense, these bacteria have begun to produce these things called cytophores, which are proteins that actually can basically steal the iron back off of these iron-binding proteins produced by humans. And that's just because they have a higher affinity to the iron than the iron-binding proteins do that we produce. Another type of antimicrobial proteins are called antimicrobial peptides. So these are short proteins, 15 to 20 amino acids long, and these things can insert themselves into the membrane of prokaryotes, bacteria, and actually kill them. So examples of these are defensin, which are in your skin, meganins, and protegrins. They also have some other functions as well. So scientists are trying to use these peptides for therapeutic drugs because if they can control them, they can use them to fight off infections or do other things that, for that matter. Okay, so we've seen this before. We saw this at the beginning, but let's just kind of run through it one more time. So we had innate or non-specific immunity, which was broken up into the first line of defense, which was barriers, physical barriers, chemical barriers, and just genetic makeup. Then we had the second line of defense, which were cellular. Okay, these are cellular and chemical systems that are produced from cells, right? Includes phagocytosis, inflammation, fever, as well as antimicrobial proteins that we just finished with. And so what we're going to get into next chapter is acquired or specific immunity, which is the third line of defense. And this is memory. Okay, we have active or passive. Active is produced from an infection or a vaccine. Passive is produced from maternal antibodies through the breast milk or through the placenta. And they involve B cells, T cells, and the other cells that are produced from the B and the T cells. Okay, so put the steps of phagocytosis in the correct order. Engulfment, killing and destruction of bacterial cells, phagosome formation, adhesion of bacteria, release of residual debris, chemotaxis by phagocyte, and phagolysosome formation. And the order is chemotaxis by phagocyte, adhesion, engulfment, phagosome formation, phagolysosome formation, where the lysozymes bond to the phagosome, destruction of the bacterium, release of the residual debris, and one other thing that isn't in here that we'll talk about next chapter is antigen presentation. And that is the first half of host defenses, first and second lines of defense. So next chapter we will talk about the third line of defense, and that is the end.